After the last speaker, we have this round of discussion and questions. So the last speaker of this session is Peter Wick. At my age, I always need glasses. Dr. Peter Wick, and he is from Switzerland. Welcome. And you are from the Empa Institute. And this laboratory has the name Particles Biology Interactions. So actually that's highly needed because we inject, we see a lot of things, we may image things, theranostics or measure, but what is actually going on with our particles in the body? What is the exact in vivo fate at the level of blood, cellular interaction, tissues, and in the cells? I think that's your field. Please take the floor.
Thank you very much for the kind introduction. It's a great pleasure for me to be here, and I would like to thank the organizer, in particular Beat and Patrick, for giving me the opportunity to share our newest results of a project we started now 1.5 years ago, roughly, to establish patient-derived glioblastoma tumor models. I think in this audience I do not have to introduce too much what glioblastoma is. It's a malignant brain tumor of glia cells. There is a standard therapy in surgery, radiation, and chemotherapy, and the survival rate is not that good. And we are still have pure treatment outcome, and we, as an Empa team, try to contribute with novel model approaches to boost the preclinical research, to be more explorative and to have good models which provide also some meaningful clinical outcomes. So where we are with the preclinical model research, I think I do not have to introduce here the xenografts, which is definitely the gold standard with high clinical outcome. But there are some drawbacks also mentioned here. And on the other side, we have the in vitro biotechnology, did a lot of progress the last decade, in particular the last years, in having the capability to mimic dynamic microenvironment, provide, if smartly done, throughput capability, and smartly done, you can also do that in pure human-based environment. But still, there are also some gaps, and what our aim is actually to combine these two worlds together and to develop a patient-derived in vitro model capturing the glioblastoma complexity. And here, I'm very glad that recently a very nice paper was out describing how to do patient-derived organoids, tumor organoids, which helped us a lot in progressing our ideas. So what is our aim? We want to go for this organoid generation according to the Jacobs paper. Then, and I was happy to have the previous speaker to highlight how important is the vascularization. So that is actually our aim, to combine these organoids with the vascular system, and this using the chip technology, which hopefully provide us the appropriate mimicking environment so that we can put everything together in having a novel cancer or novel model for exploring novel cancer therapies. So this is how it looks like, the journey to get this organoid. So we teamed up with the next door hospital we have at the Kantonsspital St. Gallen. Very gladful that Professor Marian Neidert is actually willing to give access to the operations theater of our PhD students, that they can get the biopsy directly during the surgery. And he is using this 5-aminolevulic acid treatment in order to identify where the tumor is, and according to that, we can actually differentiate between the different regions of this glioblastoma tumor. So what we did, or what we did together with the colleagues of the hospital, is that we get 3 types of biopsies, so from the tumor core, from the contrast enhancing area, and from the peripheral area, in order to explore which area has the most predictive power and how these organoids will evolve over the cultivation process. Thereafter, we travel back to Empa and follow the Jacob paper in order to get these organoids. And you see down our verification that this organoid formation during cultivation becomes nicely spheroid. And this is the starting point actually for the next steps. So we did a lot of characterization according to the Jacob paper, but also together with the pathologists at the hospital in order to have exactly the same endpoints analyzed as they would do with the primary tissue in order to retrospectively have the best possible comparison strategy to understand what we produce as an organoid in order also to define where are the strengths of this model and where are the limitations. And you see already here as a glimpse that patient to patient we have high variability. This is not new to this audience, I guess. And this is what we want to deal with and to anticipate in order, hopefully in a long-term perspective, having enough biopsies so that we can also do some advanced statistics on it in order to describe and quantify this heterogeneity. Here is just one example how we assess the proliferation rate. By classical staining, I think these are all well known in this audience. What we also try to do is actually to go a step beyond the classical pathological analysis. There is strong evidence in the literature that the nuclear morphology can help to distinguish the different cell types, but also the different state in the dynamics of the different cells. So with our two very talented PhD students, in particular Selina, was actually able to stain these organoids by a DAPI in a very homogeneous way so that we have clear view on the nucleus. And if you go closer into, we can actually zoom in and we see the different morphology of the nucleus. And here we can actually, with some image analysis system, we can actually start to describe the nuclear morphology parameters like roundness, the ellipse axis, and so on. And we can actually very nicely quantify that. Now, this is the start of the procedure. So she managed to homogeneously stain the whole organoid, so we can analyze the whole organoid. And we are now in the process to find some training data sets in order to apply machine learning to speed up this process to analyze all this nucleus. Otherwise, my two PhD students would counting the whole PhD time, and this is not the purpose of a PhD. So the next topic is actually how we get the vascularization into the organoid in a chip. So there are some chip designs which are already described, which are commercially available. This is our starting point, and hopefully in the process to use them, we may come up with our own designs, and colleagues at Empa have all the tools that we could actually introduce our own future design. But without vascularization, I think these organoids do only half of the job, what they should do. And I'm really was happy to have the introduction to see where is the entrance point, and I was not aware that the outside port is as important, so that was already good enough. I was here, thank you for that hint. How we want to do it, it's not that easy, and we have not that many patient-derived tumor organoids, so we stepped back and used some cell lines to create these spheres in order to explore which particular technique might be the most successful one to combine the organoids and the vascular tissue. This is mentioned here, but at the end of the day, we would like to find a method to combine the vascularization and the tumor organoid I showed before. So my two PhDs started actually to explore what is described so far in the literature. So the first approach is to co-culture spheroids with the vascular bed in the hope that they find each other and start to invade the tumor so that we have the vascularization. There is another approach that you generate spheroids from glioblastoma cells and you add some endothelial cells and fibroblasts in the hope with the self-assembly process that this structure were developed. Or you go for spheroid fusion. That's another approach which was described in the literature. We are now in the progress to explore which one works best for us. So you see, or maybe hard see, there are similar outcomes as is described in the literature, but you all agree probably with me that there is still room of improvement, and that's exactly what we're striving for to go to. Then last but not least, if you rely on organ on a chip, you have also to think beyond that and how this data can be processed in the future. We all know that if you complete the preclinical research, you may have to make the first step to define the dose for first in man. If you rely now on organ on a chip, you have also to provide the calculations or the rationale behind how you extrapolate out of organ on a chip the dose for first in man or first in women. So we have some ideas how we want to do so, that we have an in vitro in silico model for the prediction of the drug dosage for novel glioblastoma treatments by combining the dynamics, the PK modeling, and we have one of the PhD some background in this area. So we hope also then with the first data we have from known treatment that we can extrapolate that and provide also here a meaningful approach that we can define this dosage. So with that, I would like to thank my team. It's always a pleasure to present the data in front of a big audience, but the hard work are these persons here in particular. I would like to, now it got frozen. I wanted to, yeah. The NICE group, this is one group in my lab. In particular, I would like to mention Vanessa, a senior scientist supervising this activity. Selina and Sue are the talented PhD executing that project. And we were very glad to have substantial support from different foundations in Switzerland, believing in our concept, and also our collaborators at the hospital. And my new affiliation at ETH helps me also to get connected more into this business. And with that, thank you very much for your attention.